Hello, we are going to talk about cell transports today, how things can get into and out of the cell. We're going to start off by reviewing diffusion and osmosis. Again, the movement of molecules from an area of high concentration to an area of low concentration. Okay, so this is the movement of molecules without the use of any energy. It just naturally happens. It goes from an area of high concentration to an area of low concentration with no help. Okay, so again, we're moving from high to low concentrations. Facilitated diffusion occurs when we need to increase the rate of diffusion. And what's going to help it is the use of carrier proteins. And there are different carrier proteins that will bind to sugars or different ones that will bind to different amino acids. Okay, so we're going to have facilitated diffusion occur within our body when we need to get, like say, glucose into our body quicker. So it is used when the rate of diffusion into the cell must be speeded up or accelerated. And also, it's going to help in the ones that have a little bit of a bigger size or their charge is a little different. So it's going to allow them to go through the carrier proteins to help them get into the cell. And this is just showing you the carrier protein. It kind of looks like a hot dog bun, and things will pass through. Okay, so passive transport was no energy. Active transport is movement of molecules into the cell or out of the cell that requires the use of energy. So it's going to go in the opposite direction. It's going to flow from an area of low concentration to an area of high concentration. Okay, now it doesn't happen normally. We need to have ATP. We also need to have a carrier protein or those hot dog buns or a structure called vesicles. So we're going in the opposite direction. We need help with energy and a way for it to move. And we use carrier proteins or vesicles. And this is just showing you going from low to high, and we are needing ATP. Again, in active transport, we do need ATP because it goes from low to high, and it can be a vesicle or a carrier protein. Okay, there are two main types. We have endocytosis and we have exocytosis. Endocytosis is the process of bringing things into the cell by using a vesicle. Okay, and we have two types. We have pinocytosis and we have phagocytosis. Pinocytosis is what we call cell drinking. Think of like a pina colada. So small particles or liquid droplets are going to enter the cell through the vesicle in pinocytosis. Okay, phagocytosis is food, nutrients entering into the cell. So large solid particles entering the cell. And again, it enters through with a vesicle. So we got a kind of a pushing through of the plasma membrane, and then parts of the plasma membrane will break off, forming a vesicle for it to get through. And you can see here, either this is pino or phago, doesn't really matter, but we're pushing through the cell membrane, we're starting to break off, and this is the vesicle that we have formed. So endocytosis is the process of getting something into the cell. Okay, so the last thing we're going to talk about is exocytosis. And this is the process of getting things out of the cell by using a vesicle. So it's the reverse process. Okay, so a vesicle is in there probably containing waste. It's then going to fuse with the plasma membrane and then actually have the stuff exit out of the cell. So it's the reverse process. We have a vesicle. The vesicle is then going to enter into the cell membrane, and then all of the stuff leaves. Okay, if you guys have any questions, please see me tomorrow. Thanks.